1 second to bring up what I need to bring up. Okay. So, okay, everybody ready? Lorette, you ready?
Yeah.
Jay, you ready?
I am ready.
And Dan, you ready?
I'm ready.
Okay, you can go ahead and start recording, Tom. And then, hello everybody, my name is Sean Brown, and I'd like to welcome all of you to the Bridge webinar series today. The Bridge webinar series is designed to prepare for the next generation of big data analytics woven into transdisciplinary and intersectoral sciences, policy, and innovation, and serving as a catalyst for solution at scale to better address the seemingly intractable problems that lie at the nexus of health and wealth production, distribution, and consumption. This is a webinar series that we put together as a joint effort between McGill Center for the Convergence of Health and Economics, Johns Hopkins Global Obesity Prevention Center, the Pittsburgh Supercomputing Center, the Mission 2.0 Project, and Healthy Brains for Healthy Lives. My name is Sean Brown. I'm the Chief Software Architect for SeaBrain here at McGill University, and I'm a representative of the Healthy Brains for Healthy Lives initiative, which is an organization that seeks to build infrastructure and scientific pathways to bridging neuroscience and behavioral sciences, social sciences with advanced big data analytics and other techniques. So I will pass the baton off to Jay to briefly introduce himself and his organization.
Thank you, Sean. I'm Jay DePass, the Director of the Public Health Applications Group at Carnegie Mellon's Pittsburgh Supercomputing Center. Our group applies high-performance computing resources to non-traditional applications in public health modeling, translating modeling tools to new and broader audiences for national and international public health decision support. So I'm here representing both the PSC and the MIDAS Mission 2.0 group, which is the networking and education arm of the Informatics Services Group, funded under the Models of Infectious Disease Agent Study, MIDAS, by the NIH. MIDAS is a collaborative network of research scientists who use computational, statistical, and mathematical models to understand infectious disease dynamics and thereby assist the nation to prepare for, detect, and respond to infectious disease threats. The goal of the mission group is to allow software developers from the national and international network of MIDAS research groups to work together and learn about new technologies and practices and expand the boundaries of the work that they do. So thank you all for attending, and I'm going to hand over to Lorette to introduce herself and today's speaker, Dan Belsky.
Thank you. I will keep my introduction and the introduction of the center very short. I'm Laura Dubé. The center specializes in what we call a brain-to-society approach to address modern health challenges that are tied to a complex interplay between biology and the environment that we are all creating in various dimensions. The presentation today by Dan Belsky, Genetic Discovery for Educational Attainment and Social Class Mobility, and analysis of 5 longitudinal studies. This presentation is the quintessence of the type of research we want to foster, both at the center and by our engagement in this series, is to bring together in novel way behavioral and social science with quote-unquote the hard science, and bring all the power of omics and other digital technology to see what can we do as science to do better than we have done before in scaling up a real-world solution. Dan is assistant professor in the Department of Population Health Science at Duke University's School of Medicine, and also assistant research professor at Duke Social Science Research Institute. Is work bring together discovery from the cutting edge of genome science and longitudinal data from population-based cohort to identify mechanisms that cause accelerated health decline in older age. His work very interestingly take a lifespan approach that encompasses research on cohort of children and across the whole lifespan, with the goal of understanding why, among other key questions that he is addressing, why socioeconomical disadvantaged population suffer increased morbidity in older age and earlier mortality, and to devise a strategy for a more impactful intervention. Dan, welcome. We are very much looking forward to your presentation.
Well, thank you very much. Thank you all for having me. After that introduction, the presentation today may seem a bit of a non sequitur or slightly off topic, but hopefully by the end it will become clear how this work relates to that larger goal of understanding causes of health disparities and devising new strategies to address them. I'm going to talk to you today about genetics and social class mobility, and I'm going to show you here the next slide. I thought I was going to show you the next slide. There we are. A bit of an outline of what I'm going to get into. So I'll talk to you first about why I think we should be interested in genetics and how we should go about studying them. I'll talk specifically about the method polygenic scoring to devise measurements of the genome for analysis, in particular in the social and behavioral sciences. And I'll go on to talk about the application of that method within the social sciences and about GWAS discoveries for education, which is what the focus of the talk today will be about. And then finally, I'll get into the actual empirical meat of what I'm going to show you, and then we'll do summary and conclusion. So I like to start the talks that I give to interdisciplinary audiences with this kind of question, which is, why should we study genetics? And then as a related point, how should we go about doing that? You're here, so obviously you've answered that question to some extent for yourselves, but this is the answer that I give when I'm talking to social scientists about why we should study genetics. And the bottom line is that everything we study in the social and behavioral sciences is what those in genetics would call heritable. In other words, traits, behaviors, outcomes tend to be more similar among people who are genetically closer relatives. So siblings are more like one another than are half-siblings. Monozygotic or identical twins are more like one another than are dizygotic or fraternal twins, and certainly blood relatives are more similar to one another than are adoptive relatives. There's nothing deterministic about the relationship between DNA and these various outcomes. The implication of this broad heritability of human behavior, health, and social attainment is that genetics has something to do with them. And as an etiologic factor, generally DNA is kind of special because it comes before everything else. DNA is fixed at conception, and it remains unchanged across the life course, and that provides us with a somewhat unique quantity for conducting analysis of social and behavioral phenotypes. The idea here is that by studying genetics, we can learn something about pathways that give rise to social or behavioral outcomes that we'd like to see either more or less of. And by understanding those pathways, we can devise more effective interventions to modify human lives to prevent the bad and make more of the good. Okay. So with that background on why we should study genetics, the question then becomes, all right, so if we're going to study these genetics, which genetics is it that we would like to study? And for those of you who've been around for a little while, you can remember there was a time when the way we did this was something called the candidate gene approach. And the idea here was to try and essentially reverse engineer the biology of the thing we wanted to study, to come up with some proteins that we thought were important in a biological process that produced that outcome. We would then find the genes that encoded those proteins, find variants in those genes, and then conduct analysis of those variants to investigate our hypotheses. That candidate gene approach was awfully slow going, in part because it turned out we weren't able to make very good guesses about which genes should be most relevant to the outcomes we wanted to study. And so with the sequencing of the human genome and advances in technologies to assay genomes, we have moved to a more hypothesis-free approach, at least for initial discovery. And the idea of this hypothesis-free approach, which you probably heard about as a genome-wide association study, or GWAS, is that we're going to leapfrog our existing knowledge of biology. We're going to conduct comprehensive testing of all the variation we can measure, and we're going to let the data tell us which of these genes are more important. So the design of a genome-wide association study you can see here, and the target of analysis you can see on the left-hand side of your screen is a cartoon illustrating a single nucleotide polymorphism. So this is a single letter change in the human DNA sequence, one nucleotide, and polymorphism means that it's relatively common, so you're going to find it in at least 1% of the population. And what the genome-wide association study does is it takes a comprehensive survey of these SNPs, single nucleotide polymorphisms, from across the genome, measuring between 500,000 and several million. And for each of these SNPs, it essentially compares the frequency of the rare variant, the less common variant of the SNP, in people who have a disease, the cases, and in people who don't have a disease, the controls. Or you can imagine scaling that to a quantitative phenotype. The idea is it's just a question about whether these variants are more common in people who have the trait or condition that we're studying than those who don't, and that's being done over and over again millions of times, and a statistical correction is applied to account for multiple testing. So it's a brute force approach. It's extremely simple. The analyses typically involve very minimal numbers of covariates, but as a discovery enterprise, it's in fact borne a surprising amount of fruit. And so what I'm showing you here are two different graphics depicting the growing success of the genome-wide association study method. On the right-hand side, you can see an output of the results from the GWAS catalog. This is now a year or so out of date, but the vertical lines are essentially showing you our 23 chromosomes, and the colored dots annotating those vertical lines are genome-wide association study discoveries for different kinds of health conditions and characteristics. So the different colors indicate whether it's a cardiometabolic phenotype, a neoplastic phenotype, a neurological phenotype, an anthropometric phenotype, et cetera. And then another sort of visualization illustrating the growth over time in the volume of discoveries is shown on the left-hand side of the slide. And there are references there if you want to go looking for these, what I think are fairly nice summaries of the progress of genome-wide association studies. Now, the challenge in all of this discovery is that the individual variants that have been identified in GWAS have extremely small effects on risk for disease or influence on variation in a quantitative trait like body mass index or height. When we think about diseases, individual variants might confer an additional 1% of risk if it's a comparatively large effect variant. So in fact, we're often talking about identified genotypes associated with fractions of a % increment in risk for a disease or a trait. And that genetic architecture, as we might call it, in which very large numbers of variants collectively contribute individually very small amounts of phenotypic variance, poses a translational challenge for GWAS. Because the traditional path to translating a genetic discovery is that once we've identified the DNA sequence, we can then trace a path from the bottom up, so to speak, forward from DNA to RNA transcription to protein production and on up a biological chain of causation to the production of the disease or the phenotype we're interested in studying. And the goal of this exercise, this bottom-up translational exercise, is to identify a molecule that can serve as a target for an intervention, typically a drug. But if the individual DNA sequence variants we're discovering have such tiny effects on risk, it's unlikely that the molecule that they're going to lead to is going to be tremendously influential. And in fact, even if the path can be traced, there's now a great deal of concern about how specific that path is likely to be. So the translational challenge in the first case is that there are very few common genetic variants that have large effects. And these large numbers of variants with small effects are not clustered tightly within disease or trait-specific pathways. And there was a very influential article published last year in Cell that you can see there on the slide, sort of making this case that as we conduct ever larger genome-wide association studies, as we do this data mining of the human genome, what we're coming up with are variants in genes that are expressed very broadly throughout the body, and so intervening on any particular pathway that we identify is not necessarily going to have highly specific effects on the target that we're interested in. So when we came to this work, this challenge was already emerging as a problem. So if there was going to be public health relevance to genome-wide association to study discoveries, this classic path from identifying a DNA variant to building a drug to target a molecular process wasn't necessarily going to be the only way we were going to have to go. And so what we have been working on instead is what we call a top-down translational approach. And the idea here is to trace the path essentially backward from the phenotype, to use the knowledge that we've generated in social and behavioral sciences or clinical sciences about the natural history of a trait, a behavior, or a health problem, and then begin asking to what elements of that natural history are these genetics related. And the goal of that investigation, instead of a molecule, is a developmental stage or a process in the etiology of the phenotype that can now become the target of a non-drug intervention, an intervention like a program or a policy. So, for example, in our work on obesity, we were interested in when in development genetic risks discovered in genome-wide association studies of adult body mass index came to be associated with this pathogenic process of obesity. And obesity is a phenotype whose development begins very early in life. So people who grow up to become obese tend to be born larger. Their birth weights are higher. They then put on weight more rapidly during infancy and early childhood. And that, in turn, accounts for substantial differences in obesity risk in adulthood. And what we found in following up genetic discoveries made in GWAS of obesity, in which the studies had focused on adults, was that these genetics weren't related to how big babies were when they were born. So they were not associated with patterns of growth in utero, but they were associated with patterns of weight gain in the first years of life. And in particular, the rate of gain in weight during middle childhood proved to be a critical mediator of associations between genetic risks and adult obesity outcomes. And I think the policy implication of that observation would be that interventions to disrupt the obesogenic process that kids are facing today might most productively focus on that developmental window. So in that study and in the other studies that we conduct, we apply a particular method to summarize the findings of genome-wide association studies, because again, there's this challenge that individual genetic variants have very small effects. Another way to say that is there's no gene for X. So there's a headline out, there's a love gene, there's a warrior gene, there's an intelligence gene. There's not. There are hundreds or thousands of them, and they're more or less distributed along a continuum. The goal then is to try and summarize that continuum. And so in the figure here on the right-hand side, I'm showing you an illustration from a 2009 Nature Reviews Genetics paper, which is in fact a bit of math that Fisher published toward the beginning of the 20th century, essentially illustrating that as you add up independent quantities, you quickly approach a normal distribution. And this normal distribution happens to reflect what we believe about liability to quantitative traits and in fact, to common diseases. And we can produce this normal distribution from the results of a genome-wide association study based on some work initially done by the first three people whose photographs are shown on the page. So Peter Vischer at the top, Naomi Ray, and then Sean Purcell, whose 2009 Nature paper about schizophrenia kind of debuted this method to the broader scientific community. And the method, in fact, is very straightforward, so I'll walk you through it. Essentially, you take a GWAS, and here I've shown you on the far left-hand side of the slide a GWAS chip. This is what the product looks like from Illumina that does these chemical assays of the DNA sequence. And you essentially match the genotypes you generate from your sample with the results published for a genome-wide association study. And the figure there is what's called a Manhattan plot, illustrating associations between variants across the entire genome, the chromosomes numbered 1 to 22 on the x-axis of the figure, against their log p-values, which are on the y-axis. And so more statistically significant associations tend to place higher on the graph, and that gives it this appearance of a series of tall buildings, hence Manhattan plot. But the idea is you match the genotypes of your participants with the results from the genome-wide association study, and then you essentially use the results of the GWAS as a scoring algorithm. You count the number of phenotype-associated alleles at each SNP. It could be 0, 1, or 2. You get 1 from each parent. You multiply it by the coefficient estimated in the GWAS, and then you calculate an average across all of the SNPs in the genome. There are some different procedures that can be taken along the way, but broadly, that's the exercise. And what you get out of that exercise is this normally distributed measurement that summarizes genome-wide influence on the trait or condition of interest. That measurement is going to become the tool we use to test how genetics are influencing risk for a disease, or in the case of the study I'll show you today, are associated with social mobility. There's a caveat, and it's a pretty important caveat. So the measurements that we take in GWAS are not themselves understood to be what we call causal variants. In other words, the single nucleotide polymorphism I measure in GWAS may itself have no direct effect on the phenotype I'm studying. What we do assume is that the signal we detect from a SNP reflects signal from a causal variant. So some DNA sequence change is giving rise to this association. And that DNA sequence change is being measured by the SNP. The SNP is a proxy for some unmeasured causal variant. And it's a proxy because of something called linkage. And linkage is essentially the way geneticists talk about correlation between different sites in the genome. And so when you measure a single nucleotide polymorphism in a GWAS, essentially what you're doing is measuring common variation in the region which is in linkage with that SNP. And you can see in the figure on the right linkage plots illustrating patterns of correlation between a SNP, which is in the cholinergic nicotinic acetylcholine A5A3B4 gene cluster on chromosome 15, and is the lead hit in genome-wide association studies of smoking behavior. And what these triangles with different amounts of red are illustrating is essentially the correlation between that lead GWAS SNP and surrounding DNA sequence. And I'm showing this to you to illustrate the difference in the picture that you see in the triangle on the bottom, which shows reference genomes for European descent individuals, and the triangle on the top, which is showing a reference genome for an African descent population. And what's being illustrated here is that this particular genetic measurement, this SNP, proxies different sequences in Europeans and in Africans. And the consequence is that the effect size we've estimated for the SNP in a GWAS of European descent individuals isn't necessarily going to tell us what the effect is going to be in African descent individuals, because it's not necessarily that SNP which is the cause. It may be something else that is correlated with that SNP. And that pattern of correlation is different in different populations. So let me pause here and just ask if anyone has questions about this step, because this is an important step in understanding what we're measuring and importantly what we're not measuring.
Does anybody have any questions? We can, if you, we've muted everyone, so just to keep the background noise in check, but if you'd like to ask a question, you can either go ahead and unmute yourself and ask, or you can put it into the chat window as well, and we can just announce it to the webinar.
Just a quick question, Laurette here. From your last slides, it means that a GWAS will be done for each of the primary phenotype or the primary outcome you are interested in examining. Am I correct, Jay? Not Jay, Dan, sorry.
Yeah, so I'm not, so genome-wide association studies are conducted for all kinds of different things. I think as non-GWAS scientists, we're typically interested in one of those things or in something related to one of those things. So the polygenic score method is a way of taking information from these large-scale data mining experiments and importing it into a new study that we might want to conduct to understand something. So you get together hundreds of thousands of people to conduct genome-wide association studies of body mass index, and we can then take those results and use them in a new sample to conduct an investigation into the etiology of obesity, let's say.
Okay, thank you.
Does that make sense? Okay. So the point I'm making here is simply that you need the GWAS results have to be interpreted in a fairly population-specific way. So far, most genome-wide association studies have been conducted in samples of European descent. We're starting to see more in Asian populations, but we're limited in the kind of inferences that we can make when we want to move beyond the GWAS discovery population. And that problem, which exists at the level of the individual SNP or locus, actually gets much worse when we expand our view to studying polygenic scores, which are summaries of hundreds of thousands or millions of SNPs. And so essentially the errors in measurement that arise from these ancestry differences in patterns of linkage or patterns of genetic correlation between sites on the genome accumulate as you add more SNPs to your measure. And so what I've shown you here in the figure on the right side of the slide are a set of distributions for polygenic scores for height, stature, based on a genome-wide association study of Europeans. Now graphed for several different populations, and the key shows you the different populations. So the kind of pinkish distribution is an African descent population. The greenish distribution is indigenous Americans. You're seeing East Asians in the purple distribution. The light blue is the European distribution, and then there's a South Asian distribution in orange. And I use this because I think the juxtaposition of the European and the African distributions is very stark, and it's completely opposite to distribution of the phenotypes. So on average, Africans tend to be taller than Europeans, and yet here when we're comparing these polygenic scores, we're essentially seeing the opposite. And the point is that the distributions themselves are not comparable. And the information generated in a genome-wide association study of Europeans doesn't have a straightforward translation into a population of a different ancestry. Now, it remains somewhat of an empirical question whether within populations of a specific ancestry, the polygenic score can still capture phenotypic variance. What is certain is that it captures much less variance. So polygenic scores implemented in a population other than the one the GWAS was conducted in will be, measured with substantial error. Whether that error completely overwhelms signal or not, I think varies on a case-by-case basis. But the Martin paper is a nice introduction to this issue for those who are interested in doing more of it. I think the important thing to understand for now is that for genome-wide association studies, most analysis of polygenic scores derived from those GWAS are limited to populations of European ancestry. Before applying results from GWAS of Europeans in another population, there has to be some careful validation testing. And certainly the results I'm going to share with you today will be limited to a population of European ancestry. Okay. So even within populations that broadly share ancestry like Europeans, there are actually meaningful and detectable genetic differences. So what's being shown here on the right-hand side of the slide is a plot of principal components estimated from genome-wide SNP data from a European sample. And the first two principal components are being plotted on slightly off-kilter X and Y axes, and they're overlaid on top of a map of Europe. And what you can see is that the color coding, and if you look more closely, in fact, letters correspond to countries of origin, and there's substantial clustering of individuals plotted along these two principal components within the countries from which they're coming from. And if we believe that there may be differences between these countries in policies or culture that could causally affect our phenotype of interest, that's going to represent a kind of confounding. And it's a confounding we call population stratification. The sort of most famous statement of the population stratification problem is called the chopsticks problem, and it's a 2010 molecular psychiatry paper by Hamer and Sirota. So I've got the chopsticks up there in the top right corner of the screen. Essentially, the thought experiment they suggest is one in which you ask if you can identify genetics for using chopsticks instead of a fork. You don't pay any attention to the ancestry of your sample. You do your analysis, and lo and behold, you find these large effect variants predicting chopsticks use, and really all you found are genetic variants that tend to be more common in people of East Asian ancestry. So that could be a problem if we think about how health risk behaviors or health services or access to resources may vary across, let's say, geographic origin within Europe. We can, however, address this population stratification challenge. We can estimate these principal components from the genome-wide SNP data, and we can use them as covariates in our analysis to account for this kind of confounding. Okay. So, getting closer to the meat here. I call what we do sociogenomics. I think that there are other uses of this term, in particular as it relates to genomic regulation of social behavior, most famously in honeybees. So that's not what we're talking about. We're talking about the application of genetics within the social sciences. So I think, I want to draw a distinction here between, the fundamental question that genetics is trying to answer and the fundamental question that we're trying to answer. So I think of genetics as being a science concerned with the question of how does DNA assemble life? And in the social sciences, we have a slightly less lofty ambition. We're interested in how DNA contributes to the assembly of life courses. And I have a kind of a specific path of research in mind when I say that. So this is sort of what it looks like. We can begin with a search or discovery enterprise that asks the question, which variants should we study? And that method, as I've explained, is the genome-wide association study. What follows is that a top-down translational approach in which we use polygenic scores to conduct developmental follow-up of GWAS results to ask when and how in human development do these identified genetic differences influence differences in the outcomes that we're interested in studying. And then finally, once we have an understanding of when and how these genetic differences become manifest in human development, we can begin thinking about how environments might modify those relationships, ultimately informing the development of policies or programs to improve health and well-being in the population. My work has mostly been focused in that middle stage of developmental follow-up, and we've worked on obesity, smoking behavior, and asthma, and more recently on socioeconomic attainment. But there's a variety of references here if you want to jot something down and look up what it is that I'm talking about. Okay, so that brings us to the topic of genome-wide association study discoveries for education. And this was, to me at least, a big deal when the first GWAS was published in 2013, because this really took the methods beyond biomedical science and brought genetics squarely into the social and behavioral sciences. So there have been 2 genome-wide association studies of educational attainment conducted so far, first in 2013 and the second one in 2016. The third one should be forthcoming soon. You can see, in fact, its results were teased in a recent Nature Reviews Genetics article by Robert Plomin and Sophie von Stum. And what the figure there on the left-hand side of your screen is illustrating is the R-squared, or percentage of variance explained by the polygenic score, which early on was small and is now growing to be quite substantial. Just to clarify, the objective of this scientific exercise of conducting a genome-wide association study of educational attainment was not meant to uncover a straightforward biological path between DNA and performance in school. Educational attainment was conceived as a proxy phenotype. Genome-wide association studies, because they are data mining exercises, are very power hungry. You need extraordinarily large samples to make discoveries. And the psychological characteristics that we might think of as proximal determinants of outcomes in schooling are not measured in samples of the same size as education. So the first GWAS had more than 100,000 people in it, the second one over 300,000, and the forthcoming article more than 1 million individuals. And those kinds of samples simply weren't possible with more specific measurements of psychological characteristics. You can see on that cartoon from the Pullman and Barnstone paper, alongside the orange line showing sample sizes and R-squareds for GWAS of educational attainment, or EA, there's a blue line showing sample sizes and R-squareds for GWAS of measured intelligence. And so the progress in accumulating samples with these kinds of measures of cognitive test performance has been much slower, and the predictive power of the generated measurements is in fact much lower. So that's what we're thinking about when we're thinking about GWAS of education. We're thinking about genetics that are related to characteristics and behaviors that advantage people in their progress through school. But of course, in bringing GWAS and genetics beyond biomedicine and into the social sciences, these researchers occasioned some pushback. And I think this article from National Geographic maybe crystallizes the sentiment. And I also particularly liked this quote pulled from Ed Young's Atlantic Monthly article on this same bit of science. There are people and who are quite legitimately concerned with the integration of genetics and social sciences because of what happened the last time these two fields came together in the early part of the 20th century and gave rise to the eugenics movement. And so I think it's incumbent on researchers in this intersection of genetics and social sciences to clearly articulate what it is that they're after. I think the people who have done the GWAS of educational attainment, the Social Science Genetics Association Consortium, have in fact taken great care to articulate what the goals of their research are and what their findings do and don't show. And they have lovely FAQ pages on their website, so you can find them at ssgac.org. I've written a bit about what I'm after. I think, as I've explained to you already, in a couple of places you can find this at the Jacobs Foundation's Bold blog, at the NIH's Connector blog, and in the Harvard Business Review, where we talk about why studying genetics related to educational attainment can be a sort of productive enterprise for advancing social welfare. Again, because we're not interested in picking individuals using these genetics, we're interested in understanding the processes that produce differences in outcomes so we can devise environmental interventions that improve outcomes for everyone. We initially applied this approach in a paper that we published in Psychological Science a little over a year ago, in which we wanted to ask, when people do genome-wide association studies of education, what is it that we're finding? So do these genetics predict outcomes beyond the completion of formal schooling? We wanted to understand the kinds of behaviors or achievements that form the developmental path connecting DNA sequence with life outcomes, and ultimately what psychological characteristics mediate genetic effects on life success. And so broadly we found that in fact these genetics discovered in GWAS of educational attainment were associated with a range of advantageous socioeconomic outcomes beyond the completion of schooling. So we conducted analysis of occupational attainment, of assortative mating. So people with more education-associated alleles tended to marry partners with higher socioeconomic status. And they, in fact, were more geographically mobile. So they, in the case of our sample, which was based in New Zealand, were more likely to leave the country, often in search of employment opportunities. The developmental and behavioral path connecting DNA sequence with these advantageous life outcomes began very early. So children who carried more of these education-associated alleles tended to talk earlier. Interestingly, they didn't hit any other developmental milestones early. They didn't walk any earlier. They didn't potty train any earlier. They didn't learn to feed themselves at younger ages than other kids. But they did say their first words and begin to use sentences ahead of their peers. And they went on to learn to read faster, to form higher educational aspirations, and to do better in school, including on standardized tests. And then, of course, they went on to complete more education. And those genetic associations with this developmental path were mediated by both cognitive and non-cognitive skills. So at the very bottom of this figure, you can see some of the data from that paper, which we wrote with the very first iteration of the educational attainment GWAS results. And on the left side, I'm showing you magnitudes of associations between a standard deviation increase in polygenic score in the genetic measurement and IQ score standardized to have a mean of 100 and a standard deviation of 15. So the y-axis is showing IQ points or 15ths of a standard deviation in cognitive performance. And it's showing that the magnitude of association grows from age 3 roughly through early adolescence. The right side panel is showing that same data in a slightly different way, in which the same test was administered to participants when they were 7, 9, 11, and 13 years, and their scores were age normed so that a value of 8 is the average score for an 8-year-old, a value of 10 is the average score for a 10-year-old, and what I'm illustrating in this little figure is that kids that had higher polygenic scores, who carried more of these educational associated alleles, tended to grow apart from their peers in this metric of mental age. So genetic associations with cognitive performance were apparent early on and grew larger over time. But we also found evidence that these same genetics were associated with non-cognitive skills, with the ability to delay gratification and act planfully, called self-control, and with their ability to get along with others, which we in the paper called interpersonal skillfulness, and that collectively cognitive ability or cognitive skills, self-control skills, and interpersonal skillfulness mediated genetic associations with life outcomes. So, and in fact, there have been some other follow-up studies, so I just want to flag this one for you. I understand that there may be economists on the call, and if you don't know about these guys, you should check them out. They've got a paper in the gray lit right now, slowly making its way through the econ literature review process, trying to study how these genetics may influence investment behaviors and give rise to wealth disparities in older adults. I think a particularly attractive finding from their study is the observation that the kind of retirement plan you have makes a difference in how much your genetics matter for wealth differences between people. And finally, I would point to Paige Harden at the University of Texas at Austin, who I think has done the best job of writing about the moral implications of genetic discoveries for educational attainment, intelligence, and so forth, on this Genetics of Human Agency blog, which is a project run by Eric Turkheimer and some collaborators, bringing together social and behavioral scientists with philosophers to think through the implications of genetic influences on human individual differences. Okay, so that's a long preamble, and at this point I think I'd like to get into the content of this new analysis we've done, but I'll pause again and ask if there are any questions that people want to bring up before we get into the data.
Again, letting everybody know that if you'd like to ask a question, please go ahead and unmute, or feel free to put it into the chat window, and we'll relay it through the audio. Okay, Dan, it sounds like we're good to go. Go ahead.
Okay.
Yep.
Okay, so again, in this project, we were interested in asking the question: what does it mean to discover genetics associated with educational attainment? And so one of the things that could happen when one was to do GWAS of educational attainment, so essentially compare allele frequencies between people with more and less education, is it possible that you can find essentially a record of assortative mating among social elites? So people with wealth and resources tend to marry other people with wealth and resources, and they tend to make sure their kids get well educated. That's going to lead to some degree of genetic similarity within this well-educated population. And our genome-wide association studies and other analyses try to control for population stratification differences, and it's pretty clear that we can address those at the level of broad categories of ancestry, like which country in Europe a person's ancestors were from. But whether those methods are sufficiently sensitive to capture this kind of assorted mating gradient, I think is a lot less clear. So alternatively, what we'd like is that these genetics that we've discovered, rather than simply being this record of social history, are direct influences on traits and behaviors that contribute to social and economic success. So that's the question that we're facing at this point. Are we, in fact, identifying a genetic record of a social history, or do we have genetic antecedents to important individual differences that we might be able to shape in order to improve people's outcomes? And so a simple way of asking this question is what we're calling a social mobility design, essentially to ask whether genetics predict change in social class within a child's lifetime. The idea here is we can model adult socioeconomic attainment as a function of socioeconomic position in childhood, the genetics, and relevant control variables. And we're going to do that in data from 5 cohorts in the United States, United Kingdom, and New Zealand. Here are the studies. They reflect birth cohorts ranging from the beginning to the end of the 20th century, with follow-up at various ages in the different cohorts. The ERIS cohort followed into, I guess what we call these days emerging adulthood, the Ad Health study, young adulthood, the Dunedin study, midlife, the Wisconsin Longitudinal study, and the Health and Retirement study in the United States. Into later life. And we're able to analyze outcomes in terms of education in young people, in terms of occupational attainment in midlife, and in terms of wealth accumulation in late life. Broadly, this is the sort of analytic design that we're applying. These are the different cohorts. And our control variables are essentially intended to screen out nuisance factors that may be associated with either people's genetics or their attainment, but which we don't think are important to the relationship between those two things. Because these samples have different kinds of designs, the controls are slightly different. So in the ERIS study, which samples siblings, we're clustering standard errors at the family level to account for non-independence of siblings. In the Ad Health study, which includes some siblings, we're doing the same thing. We're also controlling for the birth year of the participants because they were enrolled at the same time, but they had different ages at the time of enrollment. And they were also sampled within schools, so we're going to use school-level fixed effects to account for any variation that may arise from either the school a person attends or, more broadly, the school district that they're living in. The Dunedin study is a population representative single-year birth cohort, so we don't have any of these design features to account for. The Wisconsin Longitudinal study, again, we have information on the schools they attended. They vary in their birth cohort, and we have siblings. The Wisconsin Longitudinal study is also initially a study of high school graduates, and so we control for whether the participant in that study was a high school graduate or was sampled as the sibling of a high school graduate. And then finally, the Health and Retirement study is sampling households, and so where we have 2 individuals in the same household, we're clustering the standard errors at the household level. And you can see on the outcome level, we've got various kinds of outcomes. I'll talk briefly about them as we cover them in the presentation of results. These are the sample sizes that we're working with. They're not large, certainly by the standards of GWAS, but I think from the perspective of this kind of sociogenomics or translating genetics into the social sciences model, we have fairly comprehensive data, and importantly, in some of our studies, we have sampled the siblings, so we can conduct analyses that fairly confidently rule out social history as a confounder of genetic association. And we even have mothers and children in one of our samples, and I'll talk you through how we're going to use that information. So I'll show you results for the ERIS longitudinal study first, and then I'll get into the other cohorts. This is a distribution of educational attainment at age 18 in these kids. So that, again, there's about 1,900 twin children, about 50-50 monozygotic and dizygotic pairs. And what we find is that, as expected, kids who carry more of these education-associated alleles tend to complete more schooling. And so schooling here is denominated in general certificate of education level. They could have achieved no GCSE level. They could have achieved a level 1, which is a pretty minimal qualification, a level 2, which was something along the lines of a high school diploma level qualification, or a level 3 qualification, which is a pre-university qualification in the United Kingdom. You see about half the sample achieved that level 3 or A level qualification in ERIS. That each standard deviation increase in the polygenic score of a kid is predicting about 0.2 standard deviations in their educational level. For this categorical outcome in the figure here, the educational level is on the x-axis, and then I'm showing you the average polygenic score in standard deviation units on the y-axis. So you can see the kids who achieve no GCSE level have substantially below average polygenic scores. Those who have a level 3 have above average polygenic scores. But it's also the case that these kids' genetics are related to their social origins, which are an important determinant of kids' educational outcomes. So here I'm showing you essentially that same figure, but this time graphing the child's polygenic score against their parents' educational level. And you can see essentially the same pattern of association. When we conduct that simple covariate adjusted regression, we estimate about half of the genetic association with children's attainment is accounted for by differences in the educational attainments of their parents, and we get roughly the same result in our sibling difference design. So here we're just using about half the cohort who are dizygotic twins. The monozygotic twins have identical polygenic scores with no variation, but the sibling difference model here estimated from the dizygotic twins is asking what difference in education from my co-twin should I expect for each standard deviation difference in my polygenic score for my co-twin? And so we see that the sort of what the family brings to the table is a significant part, although not all, of the genetic association with attainment. So then the question that we're faced with is what does this mean? How should we interpret an attenuated genetic effect in the presence of control for a shared social environment? And we're aided in this interpretation by a recent paper published in Science by Kong and colleagues, in which they had this very elegant design in which they observed genotypes of both parents and their children. And they were able to ask in that study whether genotypes that were carried by the parents, but they were not transmitted to the child, so a genotype that a parent has but that the child does not, whether those genotypes predicted the child's educational attainment. And in fact, they did. And they called this effect essentially genetic nurture. So what's being illustrated here is a path between the parent's genetics and the child's socioeconomic outcome that isn't mediated through the child's genome. It's an environmentally mediated path. And the way we can estimate that in the ERIS cohort is by measuring the genotypes of the children's mothers and then testing how those genotypes predict the child's own outcome before and after we control for the child's own genetics. So this is the simple model, and these are the results where we see that the child's mother's polygenic score predicts the child's educational outcome about as well, in fact, as the child's own genetic score does. And that when both of these scores are entered into the model, we can see an attenuation in the maternal genetic effect, but the residual remains substantial, indicating the presence of what we would call a social genetic effect. So an effect of the mother's genotype on the child's phenotype, not mediated by this direct process of genetic transmission. So again, the suggestion here is that these genetics are influencing behaviors or characteristics in parents, which in turn have an effect on the outcomes of their children. Okay, and in fact, we can explain most of this apparent social genetic effect by a measurement of the parent's educational attainment. Now, I think that's a proxy for a wide range of other outcomes, but it gives a sense of how this social genetic design within a family can be used to explore sort of social and behavioral mediators of these genetic effects. So what is it about having a parent with a higher polygenic score that is helping these kids get ahead, regardless of what their own genetics are? Okay, and in replicating this design in our other course, we don't have genetics on the parents, but we can at least ask what is the effect of accounting for the social circumstances that a child was born into, the educational levels of their parents. And what we see is pretty consistent evidence that knowing something about the education of the parents a child has accounts for a portion of what we might estimate as a genetic association with their educational attainment. And I think the message here is, on the one hand, it's clear that these genetics are more than just a record of social history. They predict outcomes for children, even after we know what kind of family they come from, even when we compare genetic differences between children in the same family. But it's also the case that when we measure a child's genotype, we're capturing more than just the direct effect of their genetics on their own biology and behavior. We're capturing some features of the social context they grow up in. Okay. So the next step in our analysis was to move beyond educational attainment and think about the kind of outcomes that more directly shape the environments that people live in. And we first looked at occupational attainment, which we measured using this socioeconomic index score approach. You may be familiar with Duncan scores or Warren and Hauser scores. We're actually using tables produced by Warren and Hauser to score the U.S. Census and Statistics New Zealand's similar algorithm to score data from the New Zealand study. But the way you can sort of broadly understand this is using the MacArthur ladder cartoon on the right-hand side of the slide. We're interested in rank ordering social classes, where the top of the ladder reflects better off people, and the bottom of the ladder reflects people who are more disadvantaged. And to figure out what the rungs look like, essentially data on income and education from job holders in the U.S. Census are used to produce a score for that job. The participants in these studies tell us what job they have, we match the score, and that becomes the measure of their occupational attainment. We're measuring the social origins of participants in these 4 different studies based on a panel of indicators that are roughly similar across studies but not identical. The family's income, education, the jobs that parents hold, and then when we have them, measures of social disadvantage. In the New Zealand study, we actually have exactly the same socioeconomic status measures on the parents and the children. So that's how we're going to measure the social origins that people come from. And the analysis we're going to produce then is going to ask how people with more of these education-associated alleles tend to do once we take account of where they start. And so these are data from the National Longitudinal Study of Adolescent to Adult Health. This is a study that enrolled adolescents in the 1990s and has followed them up. These data were collected in 2008 when the study members were in their 20s and 30s. And you can see that we have a clear, our measures are performing reasonably well. People's social origins are associated with their socioeconomic attainments. Their genetics are correlated with both their social origins and their attainments. But after we account for where they start in life, their genetics are still associated with their educational attainment, and we find that same association in a sibling difference model. So just to walk you through the graph, I'm going to show you a few of these, but what's been done here, just for purposes of illustrating the association, is the study members have been divided into 3 groups. The low SES group are kids whose social origins are in the bottom quartile of the Ad Health distribution. The middle SES kids are those whose socioeconomic origins, the social status of their families, is in the middle 50% of the Ad Health distribution, and high SES in the top quartile of the distribution. And you can notice the clustering of the dots. Clearly, kids who are born into better off families tend to end up better off by the time we follow them up in 2008. That's the first point I want to orient you to. The second point I want to orient you to are the box plots at the bottom of the graph. And those are illustrating the distribution of polygenic scores for kids within that social origins category. So the vertical gray line that runs through each of these plots shows the cohort mean polygenic score. Here is the standardized score, so the mean is 0. And you can see in the box plots that those distributions for the low SES kids are shifted to the left, and the high SES kids are shifted to the right. In other words, there's a gene-environment correlation between kids' polygenic scores and their social origins. But finally, if we now look at the red regression line, we see this consistent positive slope in all three categories. No matter what circumstances you're born into, having more of these education-associated alleles predicts upward social mobility. And we can see this again for participants in the Dunedin study. Same pattern, slightly more pronounced gene-environment correlation in Dunedin than in Ad Health. In the Wisconsin longitudinal study, and here in the health and retirement study, where our outcome is wealth. We've applied an inverse hyperbolic sign transformation to address the skew, but here we're looking at wealth. And so collectively, across these studies, we see consistent evidence that genetics are associated with long-term life attainments, that they are also associated with social origins, and that net of these social origins, there is this residual genetic prediction, this genetic association with upward social mobility, with moving up the social ladder relative to where you started in life. Now, at this point, people are often interested in whether the slight differences in these slopes that you can see looking at these plots are statistically significant. So is there some kind of difference in returns on genetics depending on the social class of your family? So we've done this analysis, and I think the results are fairly straightforward. So they're not consistent is the answer. So we can see in the Ad Health study and in the Dunedin study in New Zealand, we see that kids who are born into slightly better off families tend to enjoy slightly stronger returns to their polygenic scores. The association is slightly stronger for the high SES born kids than for the low SES kids. In contrast, in the Wisconsin study and in the HRS, the opposite is true. And so I would be inclined to interpret this largely as a null result for a test of the hypothesis that polygenic score associations with attainments tend to be stronger or weaker in kids depending on their social origins. That comes with the caveat that there are substantial cohort differences between these four studies. And so it may be the case that this pattern is in fact meaningful, but I think absent specific identification of those features that drive this difference in results, you know, the conservative interpretation is that we don't see a clear association between the social origins that children were born into and how their genetics relate to their attainment. Okay, here's a summary of the effect sizes across our analyses. The navy blue dots are showing the association with attainment. The brighter blue dots are showing the associations on the left-hand side of the graph after adjustment for social origins. So those are our social mobility effect sizes. And on the right side of our graph, you're seeing the sibling difference analysis. And the reason that the navy blue dots are appearing on the family sample analysis is that we have slightly different sibling samples than our general samples, and so the effect sizes in the siblings can be slightly different than they are in the full sample. So summary and conclusions here. I think the first important observation, building on the initial identification of genetic associations with educational attainment that we're offering here in this study, is that the education-linked genetics identified in this GWAS are correlated with childhood social class. Secondly, independent of this gene-environment correlation, education-linked genetics are predicting upward social mobility within a single lifetime. And we see consistent evidence of that whether we're measuring educational outcomes, occupational outcomes, or wealth accumulation. Then finally, that these genetic associations with attainment are modestly attenuated by controls for social origin, and consistently when we use a stronger design of comparing siblings in the same family. So although we can conclusively rule out social history confounding as an explanation for genome-wide association study results for educational attainment, we're also finding some evidence that the genetic effect that we're estimating is more complicated than a simple sort of biological process beginning in a child's DNA sequence, extending onto their behavior, and then through to their socioeconomic attainment. Okay, so the conclusions that we're going to try and draw here are first that social environments have genetic etiology, and that when social scientists are studying the social environment, there is this genetic component that has to be considered. But that these genetic etiologies are complicated. They comprise direct genetic influences on traits and behaviors that shape life choices, and also indirect, or what we might call social genetic effects, that arise, as we showed, from parents, but we can imagine a range of other people who will surround children that have these higher polygenic scores, because to the extent that a child's genetics predict their own socioeconomic success, we can imagine that those genetics are now correlated with a wide range of environments that that person's children will grow up with. So not only do your parents have different kinds of resources, different kinds of skills and abilities, but so do their friends, so do their neighbors, so do your classmates, and so forth. And finally, the suggestion of these indirect effects is that there are developmental and social processes that act to mediate at least portions of genetic associations with attainment and mobility, and that these processes represent potential opportunities for intervention. Okay. Rather than getting into next steps, because I know that we're a little over time here, I'll go ahead and take questions if that makes sense to people. Is that all right?
As usual, Dan, I always have the first question. Thank you very, very much for this excellent talk that brings us to the 21st century research on this nature-nurture interaction. I have two questions that build upon your work in looking at the polygenic score as predicting of educational attainment. And in moving from candidate genes to a dopaminergic, to a polygenic score, to what extent could we, in future research, tap more into the various pathways that links the polygenetic score to educational attainment? If you look at your three-factor cognitive, interpersonal, and I think it was impulsivity or something like that, they are all tied to brain processes for which, when we were when we look more at candidate genes, it's kind of more implicit that we look at the various intermediary pathways between the genes and the outcome, whether this outcome is education and disease. That's my first question. The second question, I also liked very much the Kong et al. Article that was featured in the New York Times. And when you talk about this, when they talk about this genetic nurturing and the effects of the genome of the parents beyond the one that goes through the gene of the children, this effect is also, I believe, mediated by various behavioral and other pathways that could be better specified as we move forward. So if you could comment maybe on those two aspects.
Sure, very happy to, and thank you. Those are exactly the questions that I would have hoped for, so I really appreciate it. And thank you all for bearing with me. This is the first time I've ever given a talk to my monitors. Before I answer Laurette's great questions, I just wanted to first acknowledge my collaborators and funders, which I was remiss in not doing at the end of the talk. Ben Deming at Stanford, my very close collaborator on a lot of this work, Jason Boardman, Kathy Harris, Dalton Connolly, Jason Fletcher, Candace Odgers, Terry Moffitt, and Avshalom Caspi, Jeremy Fries and Pamela Hurd from the Wisconsin Longitudinal Study, and Robbie Guido and Jasmine Wirtz, of course, the Jacobs Foundation and the National Institutes of Health. Let me also acknowledge the deep debt that all of this work owes to Sandra Skar and those little causal graphs that you saw sprinkled throughout the talk that I didn't address because I'm not sure quite how to talk through them without pointing with my arms, which you can't see. All were very much prefigured by Skar and McCartney's 1983 paper in Child Development that laid out a lot of the logic that we're only now catching up to in the molecular genetic world. Okay, so Laurette's two questions were, first, can we get better at linking genetics with biology, and in particular with the biology of the brain within the context of this GWAS-based polygenic approach? And I'll take that one first. I think, in principle, this is very much what we'd like to do. But the challenge comes from this potential on the genetic model that I alluded to at the beginning of the talk, and I'm just going to throw that article up again. It's Boyle and colleagues. It came out in Cell this past year. And it's true that if you look at genome-wide association study results from GWAS of educational attainment, you in fact find substantial enrichment for SNPs that are located in or near genes that are expressed in the brain. But you also find genetic associations that arise from sequence that's in or around genes that are expressed broadly throughout the body. And so I think our capacity to annotate DNA sequences to specific biological functions based on our current knowledge is extremely limited. That said, I think I agree that it's valuable to work toward sort of closer specification of the biological pathways that connect DNA sequence with outcomes. And certainly, to the extent that we see genetic associations with education and adult socioeconomic success are mediated by cognitive function, interpersonal skills, self-control, that all points us to the brain. I think hopefully what we could do then is continue working with the same polygenic score, the same global summary of genome-wide association study results, and begin looking at how those genetics are related to the structure and function of the brain, perhaps structural and functional features that we hypothesize should be related to these cognitive and non-cognitive skills. We're in fact doing some of that right now in a collaboration with Ahmad Hariri and his group. A student named Max Elliott is leading that, and we hope to be able to share the results of that analysis in the next several months. So I think there is the potential to use polygenic scores to try and dive a little deeper into the biology, but I am skeptical at this stage of our ability to specify genetic measurements that are more closely tailored to biology. I think we may see in the relatively near future some statistical models that help us do that. As we get larger scale brain imaging studies and begin to conduct GWAS of brain imaging studies, a method called MTAG, there's a Nature Genetics paper, the first author is Turley, T-U-R-L-E-Y, came out earlier this year, which is designed to combine results from different genome-wide association studies, essentially to leverage the statistical power that you can generate with very large sample sizes for which you've measured very coarse phenotypes like educational attainment, essentially to generate good priors that better power investigations of more refined phenotypes. So, for example, if you're interested in how the degree of gyrification of the cortex might be related to cognitive test performance or something like that, you might imagine small-scale GWAS of MRI-based indices of gyrification. Those results could be combined with results from larger-scale GWAS of education or cognitive test performance, and from that, a genetic measurement might be refined that you could argue is more specific to this particular neural phenotypic intermediary between DNA and behavior. But I think that's complicated, so I guess I'm not sure that's an entirely satisfying answer to your question, but that is how I'm thinking about the problem, at least these days, which is we don't know enough about the genome yet to a priori say these sequences should be interesting and those ones should not be interesting. But we are in a position now to use data to begin more closely investigating neural processes that could mediate genetic associations with educational attainment, cognitive performance, et cetera. The second question was about the potential to elucidate social and behavioral processes that are mediating these effects of genetic nurture, or what we're calling more broadly the category social genetic effects. And I think this is absolutely an exciting direction that social sciences can take. Obviously, sort of an ideal setting in which to conduct these studies is one in which you have genotypes measured for both parents and children, along with observations of family processes and so forth. We can also imagine this in the context of peers or spouses, wherever we might want to go with it. But I think simply looking at how genetic differences between people relate to differences in their behavior, for example, parenting behavior, can be a powerful step toward specifying this mediational path, as Laurette was suggesting. My colleague Jasmine Wirtz is now working on how genetics discovered in GWAS of educational attainment may relate to parenting behaviors as an effort to understand some of this social genetic effect that we're observing. And so I'm very enthusiastic about that line of inquiry.
Thank you very much. Just to comment that you are thinking about those two questions exactly in the same way and with the same pragmatic realism that our network has been looking into it. So, Sean, back to you for other questions.
Sure, yeah, thank you. Since I was a little muted, so thank you, Dan, for a wonderful talk. We have a question in the chat room from Andre Portella. As polygenic risk scores are based on the original GWAS, selecting SNPs that remain significantly associated with the condition in large populations despite environmental differences. The lower the PRS-P threshold, the less likely that you'll find a PRS versus environmental interaction, because the GWAS by design selects SNPs effects that do not vary with the environment. How do you reconciliate PRSs and G by E studies?
Okay, so thank you. So the question is, given the way we design polygenic scores, haven't we almost by design excluded gene-environment interactions from the equation? And I think this is a question that a lot of people in the social and behavioral sciences have been struggling with. I think the observation is a fair one. It is the case that genome-wide association studies pool people across a wide range of environments, and that the effect sizes that then feed into the design of a polygenic score are average effects across a range of environments. That said, I don't think there's anything about that particular feature of the GWAS that precludes a G by E. Environments that are perfectly balanced in a GWAS sample, it's true, will not be the kind that you can detect gene-environment interactions with. But I think it's also the case that if we imagine environments that are relatively rare, or on the other hand, very common in the GWAS population, there's no reason that you might not detect an interaction between the polygenic score and that environment. So for example, if you're thinking about variation in educational opportunities, or if you're thinking about more extreme examples like childhood malnutrition or something like that, I think those are the kinds of environments that may modify the genetic effects that we're detecting in GWAS. And in fact, there's now evidence of this kind of heterogeneity in genetic effects within a GWAS from studies that compare genetic correlations between different samples that are being pooled in GWAS. So we find that the specific genetic correlates of a phenotype aren't the same in one sample as in another. I think the question is, can we translate that to a polygenic score by environment interaction, and is there any evidence that that can work? I think we don't have great proofs of concept there. I would point you to an article we published in the American Journal of Psychiatry last year. The first author is Deming, and in that article we test how a polygenic score derived from GWAS of subjective well-being interacts with the exposure of the death of one's spouse. And we show in that article that having more of these subjective well-being associated alleles buffers against the depressogenic effect of spousal death. These people experience kind of a lower peak in their depressive symptoms and a faster recovery. So again, long-winded to answer your question, my apologies, but broadly, I think the challenge is real. I don't think, however, this is an impossible task, and I think, in fact, in general, in the social and behavioral sciences, we're used to envisioning interactions between variables that have main effects, and that's really how we should think about the individual SNPs in GWAS and about the polygenic scores more generally, is that on average there is an effect of roughly this size in the population, but that effect could be smaller or larger. Where I think polygenic scoring and gene-environment interaction may face a steeper challenge is in essentially the requirement that the interaction operate more or less in parallel across the entire genome. And so with candidate gene G/E studies, we could imagine molecular processes in which an individual genotype interacts with a specific environment to affect a phenotype. Now what we're imagining are interactions between environments and the entire genome. And it may be the case that the nature of the gene-environment interaction that we imagine for a polygenic score needs to be a little different than the kind of specific biological synergy that we were envisioning in studies of candidate genes.
Okay, great. And there's one more question from Andre. Could the maternal social genetic effect described in your findings be in fact a maternal biological genetic effect instead? Example given, better placenta, better fetal growth, etc.
Well, I mean, I wouldn't call that a genetic effect. I would call that an environmental effect. So it's absolutely possible that the uterine environment is the mediator. I think that's a fantastic hypothesis. And particularly as we thanks to the incredible work of David Barker and his colleagues, have developed these databases that describe in detail intrauterine conditions and even various quantifications of placenta, as well as now these kids have been followed up. Certainly in the Harvard cohorts through elementary school, there's a potential to investigate this specific hypothesis. So the names of the cohorts are escaping me, but Gilman, I think it's Gilman. Anyway, Vega, I think, is the name of one of the cohorts. Anyway, they have genotypes on mothers and children. They have measurements of intrauterine growth. They have measurements of placenta. And by now, the kids should be followed up with some educational outcomes. So I think that's a fantastic hypothesis and an exciting opportunity.
Okay, great. Do we have any more? I think we have time for maybe one or two more questions. I see, Elaine, you unmuted. Did you have a question? No? Oh, hey, you remuted. Okay, so no. Are there any other questions from the floor? If anybody has a question, please feel free to put it in the chat window or unmute and ask.
Since there is still a few minutes, maybe a question that relates to life course modeling or life course influences, and to what extent, like in those 5 cohorts that you have, have those cohorts have measurement from for the whole lifespan or for just segment of it, and to what extent could we, if it's the case, to what extent could we use computational models or otherwise to get a better ability to tackle the dynamic complexity of what happened over time in terms of the change in the environment, change in behavior, and so on. So maybe a few insights on this before you close, maybe useful to the audience in this webinar.
So thank you. I think, so the question is sort of, might it be possible to knit together data from different cohorts that observe different segments of the lifespan to build a truly cradle-to-grave life course model of how genetics are related to, let's say, the accumulation of socioeconomic position and to health and well-being across the life course? I think I'm learning more about these data integration approaches. I'm collaborating with a sociologist at UNC, Claire Yang, and a psychologist, Patrick Curran, in which we're attempting to do this kind of an analysis to understand cognitive aging. So I think these methods exist. I'm still learning about them, so I wouldn't want to offer anything too specific. I think it is certainly the case that in genetics in particular, there is a degree of myopia in the conceptualization of the phenotype as a single static variable parallel to the genotype. And one of the contributions that we as social and behavioral scientists can make to the field is to expand an understanding of the phenotype to something that is, as Lorette suggested, dynamic across the lifespan. And so assembling data, whether in single cohorts, like the British birth cohorts that observe the full expansive development, or across multiple cohorts that may observe in greater detail individual segments of the lifespan, we can substantially enrich our understanding of how genetics influence our lives, and through that understanding ultimately improve the quality of the programs and policies we develop to engineer sort of better health and well-being for everyone.
Thank you so much. We may follow up on this as well. Back to you, Sean.
Yeah, so I think that's all the time we have for our Bridge webinar today. Thank you again, Dan, for a very stimulating and interesting talk. We really appreciate you joining us. Just wanted to let everyone know that while we don't have the next Bridge webinar currently scheduled, we're looking at getting our next speaker to talk about use of AI techniques in public health, looking at things like mechanisms for HIV and infectious diseases. So we'll be sending out an announcement shortly to let everybody know when that will be, and we hope you can join us then. Thank you again, everyone, for joining, and thank you again, Dan, for a wonderful talk.
Thanks very much.